Hello and welcome to the science behind black belt nutrition. In this chapter we will go through some of the secrets, you might say, how we have done our analysis, how we looked at studies and why we have been able to come to the conclusions that we have. And this has never been done by anyone else before, not within sports nutrition. And as a matter of fact, it is almost not used at all within the medical area of research. Not yet, at least. And you might ask yourself, okay, so if this thing that you're going to talk about, these statistics that we're using, if it's so good, why aren't everyone using it? Well, there might be many answers to that. First of all, they are used within the commercial companies. Not all of them, but the leading one within various different industries. And we will look a little bit of that as well, because this isn't something we have invented. And it's used, I've been working many years now within pharmaceutical, sport nutrition, and also some other engineering fields. So that is where I have learned, where I've used these methods. But they're not used very much in the world of academia, and particularly not within the medical area. And there might be different reasons for that. It's coming, it's growing. I know, for instance, that Karolinska Institute are working more and more with this, and Umeå University has probably the world leading scientist within multivariate data analysis that we will touch on here. But so far it's mainly within chemistry and engineering sciences and so on. So it's going slowly. And one reason why it's not used very much in medical research is simply that there are always some resistance to change, and there are also some information bridge that the information hasn't reached to them. Because if you look at if you develop a car and optimize the engine there, and your optimization aren't good, your engine isn't competitive, you will be out of business. So the industry has been searching for new ways to analyze data when the old ways didn't work very well. But if you publish a paper, an academic paper, and you do as they expect you to do, as they have been doing for 100 years now, it will be published, and you have your back free if you did as you're expected to do. No one will claim, even if your results aren't true, it turns out later on, you have your back free. But you can't go to your manager or to your customers and say, well, the statistics told me this, but it turns out the product we're selling is poor. It won't work. So you need to get to the right result, and you need to do it as efficiently as possible. That's really all that counts. No one really cares if you do statistics or how you do it. You just need to get good results. And that is the same for us here. We want to understand what influence training, strength, muscle mass, fat loss, and conditioning, some other health parameters as well. So therefore we thought, why not combine these industry best practice statistical methods with all the data that is found within the sports nutritions. Okay, so this is just a short background. If you want to look more on this, you can search, for instance, on Six Sigma. That is a concept that is used by many of the leading companies. And here you can find more information about it. And as I said, probably more than 90% of physicians and researchers within the medical research area, they do not know what we will show you here now. And even if you're not a fan of statistics, we will just show the principles. And we will refer to this chapter in the other chapters. It is important that you pay attention to it. It's not necessarily, if you don't want to understand, if you just trust what we say in the other chapters, you actually don't need to have a look at this. But if you want to understand it and why our conclusions might be different than others, and why different researchers disagree, you will get the answer to that here quite soon. First, some special thanks, because this isn't really done on paper, and well, it was in the old days, but so special thanks to Geometrics. This company has, we've used their software, we got to borrow them. So we used Modde for something called design of experiments. You will soon know what that is. And we used Simca for something called multivariate analysis. There are some different names on these things depending on who you talk to. And They are specialized in this. This is all they do. They have consulting and they have software to do this. And they mainly have customers within the commercial sector. They have academic customers as well, but really this has been driven from the commercial sector because they need better tools to handle their complex data, real life data as I call it sometimes. So here you can see some of their customers. They are very focused on the pharmaceutical industry, but it is used. Toyota, for instance, started to use design of experiments already 50 years ago, way ahead of everyone else. Reason were that they were behind. They started to do cars half a century after everyone else and needed to catch up. There's a design called Taguchi design, named after one of the engineers at Toyota. But most countries, most companies started to see this perhaps in the 80s, 90s, and now it's really starting to explode actually. Now everyone is starting to do this. And this also comes in when people talk about big data that is so popular to talk about, but most people don't know what it is or how to handle it. So what's the secret? What is so cryptical, mystical about this? Actually, it's quite trivial. Even if you do not have any training in this, you might say, well, of course that is the case. How else would anyone analyze data? Well, I can tell you they do it in very strange ways. Look at this example here. We have two variables. They might be anything. Might be some blood work, some test on patients we have here, minerals, vitamins, hormone levels. 40 patients. We are the doctors here now. This is patient 1, 2, and 3. And this is perhaps the testosterone level, and this is some other level of something we have tested of all these 40 patients. We have the upper and lower limit given to us by the medical lexicon. So that's nice. No one here is deviating, so everyone should be healthy regards to this test at least. What if I told you that two people here are sick and the information can be found in this data that we look at here? Do you like to have a guess which two it is? Well, is anyone closer? Well, number 29 is pretty low here, and number 29 is also pretty low here. It's not outside, but it's low in both cases, perhaps. That is what we guess on. Or maybe someone here that is high. This one, 39 I guess it is. It's not that high here, but it's a little bit higher this one. Well, how you do it, and even if you know statistics, you might be able to calculate the average here and standard deviations and all kind of things. You won't see anything more than your eyes see here. Statistics is mainly a way to make things objective so that everyone makes the same kind of judgments, but your eyes are very good in detecting patterns. So if you just plot it, if you look at the data from the right angle, your eye will tell you everything that a statistician can tell you. Maybe a statistician won't agree with it, but anyway, okay, so how do we do this then? We don't, let's say you don't see here who it is, which two patients is. It's not so obvious. Some people see it, but most don't. And there's only two factors here. If we would have 10, it would have been really complicated. But let's start with very, very simple. It can't become simpler than this. But still, it is difficult to see. And this is the way everyone is doing research. We look at one factor and calculate things on them. And then they make some conclusions. We need to look at both factors at the same time. Because that is how nature is built up. Things aren't moving independently of each other. If you move your arm, all muscles in the arm helps to do that, like an orchestra. If you look at chemistry, physics, biologically, things are connected to each other. So when you look at things individually, but in real world they're connected, you're missing a lot of things. So let's connect these two. Let's look at both variables at the same time. Now, these are red, but I am quite sure even without that you would have said, oh, okay, these two are different. I see that now. You don't see it very easily here, but you see it easy here. And if you do statistics, you can prove statistically that they are different as well. But as I said, your eye is good with patterns. You just need to look at it from the right angle, from the right perspective. Which two is it then? These two. Maybe you saw that, maybe you didn't. And this is very powerful. If you have two things you look at, combine them. This might be your own training data. You have measured things. Some days you're off, some days you're good, and you don't see why. Combine the plots and look at it like this. Maybe this explains it. Maybe you have something out here or up here that is when you're off, or the opposite, when you're really good. Then you know what factors contribute to that. But what happens if you have more variables? There are only two here. If you have three, you can look at 1, 2, A, B, A, C, B, C. You can look at all the combinations. You have three original plots, and you will then have three additional plots for the correlation. But if you have four, well, then you have more. There will be more than 4 combinations. If you have 10, then it becomes pretty hard to look at all these plots. If you're 100, it becomes hard just to calculate how many plots you can do. Well, it's not hard if you know how to do it, but it's hard to do it manually at least. So obviously there must be a smarter way to do this. Well, fortunately there is. And we have used the software, and this software, it doesn't come cheap, so that is why we are happy that we could borrow them. At least not this one that's very good that we used. They cost something like $8,000 together, or 8,000 euros together, these two. So it's quite an area. How is it done? That's quite easy to grasp, even though it might be difficult to do it manually, to calculate it manually by hand. This is how it's done. It's not so magic as you might think. Here we have 3 dimensions, 3 factors we look at, and we plot them in 3 dimensions. We can transfer that to 2 dimension by putting a flashlight over here. Here, my cursor disappears sometimes. The flashlight will then give shadows here, projections on this piece of paper. And we can position this flashlight from different points. And from some angles we will see something interesting here, but from most angles we will just see noise. Fortunately, there is a way to calculate mathematically where to position this flashlight so that we capture as much information as possible on the projection. And consequently, we don't have to worry about the noise, because these methods leave the noise behind. You can look at it if you want, but what you will see in the first picture is where you have the largest variation, where something is happening. Sometimes if there is a large data set, you need to take more than one picture. But the first one tells you the big part of the story, and the second one, the second part, and the third one. But usually just 1 or 2 is enough, even for large data sets. Well, here we have 3. Maybe you understood this. What if we have 4? I can't draw 4 here. I can only draw or imagine 3 dimensions. Well, fortunately, mathematics is not limited as our brains might be. Because you can still take 4 factors here mathematically and put this flashlight on them and calculate the projection, calculate where the shadow will be. And you can move this flashlight around so that you find out where something interesting is happening, where you have some deviation in the correlation pattern, as we just saw here in the previous pictures, like this. You can calculate where to put the flashlight to get out these kind of things when they happen. So that is how it's done. You can put in 1,000,000 factors and you get out one picture that tells you most of the story. And you can take a second one if you need more pieces. So this is an example. We have been using this throughout the other chapters for food maps and for research within sports nutrition, where we have looked at if things were done in the same way and so on. We put this flashlight on them and get simple understandable pictures. Let's look at an example here, how it can be done. Now we have taken an example that you probably know the answer to, so you can kind of confirm how these methods works. We have countries here. It's European countries. I apologize if you're from some other place and do not know how we eat in Europe, but you can probably search it. And the variables, the things we have measured in each country, or actually we haven't measured, it's some European study 15, 20 years ago, is how often or how regular they use coffee and tea and sweetener and potatoes and so on. So we have all these measurements on each country here, the variables, which is foods and countries. So countries here and foods here. If I ask you, I'm from Sweden, I want to travel to some other European country, but I'm afraid for new foods. I want to eat the same food as home. Where should I go? Where do they eat about the same food? If I ask you, I want to go to a country they eat potato, that's quite simple for you to answer because you just look at potato. You see 39% of the Swedes eat potatoes every day. Where else do they eat potato? Here you have in France and you have some places they don't eat it that often. But now I want to compare all of these so I get kind of the holistic view of it. The profile. Where do they have the similar profile? I want to look at all these at the same time and see where they have approximately the same. Or the opposite. I want to go to a country they do not eat as in Sweden. I want to take my chances here and try something new. Where should I go then? It's not impossible to answer the question, but it's very much easier if we put this flashlight on this datasheet. Let's look on the left side. What do we see here? Countries. Sweden is here. What country is eating mostly similar to Sweden? I hear someone say Denmark, and that is correct. It's quite amazing that you can answer that without having any knowledge about principal component analysis, which is the former name of what we look at here, PCA. The thing is, these plots are quite intuitive, and you will see more of them in other chapters. And if I say the opposite, what countries are eating different? Well, it's the countries that are far away from Sweden, if you measure the distance. Portugal, Italy, France. And you can see all the Nordic European countries are in the same upper right corner. And you have south of Europe down here and some middle Europe here. Okay, so this we have done to look at studies. What kind of sport nutrition studies are done in the same way, which one are different, and how many are there in the different areas. And here's something interesting arise. Okay, if they're the same way or different, why are they the same, or what difference then? Why is Sweden up here and Italy down over here? Let's go to the right plot now. You can see here that Sweden is in the upper right corner in the left plot. If you look at the same place in this plot where we have the foods, you can see it says frozen fish, crisp bread, frozen vegetables. And that means that we eat more of this in Sweden compared to the other countries in this survey. And I live in Sweden, so I know it's correct. We like this crispy bread here, the hard bread. Okay, so what do they eat down here in South Europe, Italy, Spain, Portugal, that we do not eat so much of, relatively speaking, at least not when this study was done? Garlic and olive oil. And I've been in these countries as well, and I can tell you they eat a lot of olive oil and garlic. So it makes sense. And you can easily look this up to make sure. And let's take UK for instance. I'm quite often in UK. What do they eat there? They eat biscuits and jam, according to this plot, and some other things. More than we do on average. And it's true. If you've been there, if you just look it up on the internet, they even have the jam on the biscuits. They like them so much. So you can see how we can transform this Excel sheet to something that quite easily give us information about similarities and dissimilarities and why things are where they are. We do this with studies. We instead get out protein, muscle mass, and so on here. We have done this with food maps. You see foods that are similar and dissimilar, and then you get out what they contain, what nutrients, and so on. So we worked a lot with this. And the most important thing is that it captures not only the individual variables, it pays equal attention to the individual variable as it does to the correlations with the combined variables. So if something happens over here, that is what you will get, that is what you will catch. And you never see that if you look at them one by one. So it's not even easier to look at the results, it's more powerful also. It will reveal things that you haven't seen before. Simply because the world is, the nature is put together in this way. Things are connected. Therefore we use methods that can cope with that. Here is an example of a food map, how it looks. We will go into the details on this in the food chapter. And just to say something here, we have 500 foods here, and it's still quite easy to get a picture of it. And we also had 40 nutritional values measured by the Swedish Food Agency. So it's all public data. Protein, carbohydrates, the vitamins, the minerals, and fatty acids, and a few other things. Then you can easily compare if foods are similar, dissimilar, why they're dissimilar, what food brings you something unique. We'll look more on that in the later chapters. Okay, so this is one part of the story. How to analyze data you have. The second part is how to set up experiments. So if you're a researcher, how should you do your experiments? Or if you are a self-experimenter, test different way of training and want to see which one is best for you. How should you test this? Okay, there are something to think of here as well. And this is how it's done most of the time still. You change one factor, keep everything else constant. Meaning that you don't change, you keep everything else as it is. You don't change it, you just change one thing. Then when you're finished with that one thing, you take the next one if you want to change more factors. Simply because you don't want to change one thing, and then do not have control of the others, because if they change a little bit, they can fool you. So that makes sense. Let's look what happens here. This is an old classical example, basically used in all design of experiment trainings. That is why we have it. Its chemistries are not really related to what we'll talk on in the other chapters, but it's an easy example and it's used everywhere, so we took it as well. Let's look here. We are optimizing this reaction. We are changing the time, keeping everything else constant, including the temperature. Time is changing, everything else is constant. When we change time, we get the best setting of time here, 135 minutes perhaps. We lock it here and take the next factor to change. Now we change the temperature. Increase, increase, increase. It goes down. Let's go back. This was the best spot. So we take this temperature, this time, and we have optimized our process with regards to these two settings. We pretend nothing else matters to this process. Everything else is just kept as it is, so we don't change it. It has no influence. These are the ones we need to be concerned about. In reality, of course, you have more than two usually, but we just pretend that here to make it easy. So are we done here? Have we optimized it? If we look at protein content post-workout, and we do like this, we get an answer. This is what you need. Are we finished then? Then we can go and look at creatine. Well, some of you might intuitionally know or realize that maybe things are hanging together here as well as they did in the previous example. Let's look at this in the same way. We look at them both at the same time as we did before. Here we did our test. We found the best spot here. Locked everything, moved in this direction, everything else is constant. We found the best spot here. Is this the best spot? All the blue here is the unknown area. But we happen to know how it looks in this example. That is why it is an example. This is how it looks. And if you're not used to look at these kind of plots, this is a contour plot. Here we have, it's like a mountain. We look at the mountain on the map. Here we have the highest place, the best yield. And then it goes down here. So 20%. We did our experiments. We didn't do anything wrong, really. 20, 30, almost 40, and down. This was the best one. But in this direction, now worse. Going this direction, better. Now we're down again. This is the best spot. We've optimized our process. And when you look at them one by one, it's so easy to think this is optimum. Where else can it be? When you look at like this, it's quite obvious that we're missing something here. And this is one of the major reasons why researchers disagree. Is carbohydrate good? Is fat good or bad? The settings differ, and therefore they get different results. And that is due to something called interactions. If interactions didn't exist, we wouldn't have this problem. But they do exist in nature, in physics, in chemistry, in biology, in medicine. And they can be quite profound. But since almost no one, with a few exceptions, within the medicine area. Do experiments like this. We never get the answer to what the interactions are, how big they are. And that's a big problem. You couldn't go out and start to work for BMW and say you want to optimize the cars and do it one factor by one factor, because they will be out of business. They use these methods to optimize their cars, and many other leading companies within different type of industries. And it's used in some universities, but it's so far more in the engineering science, not so much in the medical area yet. It's coming, but it's going slow. These methods are fairly new. They are mainly used by industry because they need good results, fast and accurate results. How should you do instead? Well, you need to do experiments where you change more than one factor, where you change everything simultaneously. But you need to do it according to a pattern, so you easily can back extract which factor contributed to what. With only two factors, it's quite easily. One of the designs you can use is just a square. You can see here that you already in 5 experiments have gotten up here. We start in the same place. We don't start over here. Of course, we'll get better results then. Start in the same area. The difference is that we set up it in a square like this, and the best experiment is here and the worst is here. So already in 5 experiments we have beaten the results. They ended up here, despite starting in the same place. But this is not the key, because your randomness might make you start any place here. But it doesn't matter when you do it this way, because you will see the gradient here. You will see that this spot is the best, this is worst. You can see that it's increasing in this direction. So you can do your next experiment somewhere here. If you test one factor in the time, you won't be able to calculate in which direction to do next experiments. You can't calculate this gradient. And that has to do with interactions in this case. Interactions is when two factors interact. When 1 plus 1 doesn't equal 2, it equals 22 or minus 2, depending on if they add to each other or if they prevent each other. Synergism is a word used in biology. It has to do with interactions. Let's give an example of this, because it's full of them and you know them, even though you haven't perhaps thought of them always. If you drink some alcohol, not moderate amounts, it's not really that harmful, short term at least. You take some medication that you've got prescribed. It should be safe, short term at least. You combine the two, and suddenly it might be lethal. So you take two things, 1 plus 1, you add them together, but it doesn't add up to 2. It ends up to 22. That is interactions, and it's full of them in the world of physics and chemistry and biology. It can go the other way around. Let's take you drink some alcohol again, but you get a bad batch from someone. It contains methanol. Ethanol is the alcohol we drink. Methanol is quite lethal in small doses. Just a few milliliters is very bad for you. Okay, so if I drink this bad alcohol, this methanol, which is perhaps lethal, the dose I took. Then I take ethanol at the same time, regular alcohol, then I'm like double dead or what? No, actually not. The ethanol will protect you from the methanol. If you come into the hospital with methanol poisoning, they will give you ethanol to save your life. If you get in there in time, of course. So these interactions go both ways, and they are very present all over the place. So you need a systematic way to catch them, to estimate them. Otherwise, we'll end up with this situation where one researcher tests something, maybe if it is fat is bad or whatever, and they change the amount of fat you have. Someone else is doing it out here, they say it doesn't influence anything. Someone else is doing it here and say, oh, it has a huge impact on whatever they look at. Well, that depends. What were the settings on the other factors? So that is why there's so much disagreement about how much protein to eat, how long to train, and everything. I mean, most researchers do good research. Most of them don't cheat or anything, but they draw incorrect conclusions. Well, they are correct for that particular study, but they aren't correct for the other parts of the populations. And they don't mention or perhaps say that this conclusion is correct only if you do exactly as I did. If I change something else over here, the result of my study will be totally different. They usually don't even see that themselves. Okay, so this is used by the industry. You can search on design of experiments, as you see here, Six Sigma, Taguchi design, the Toyota engineer, factorial designs, and you'll find a lot of information about it. We didn't make it up, it's used by the industry and it's used because it works. Okay, so did we do any own studies here? No, we didn't, because if we look at just one example, we did this with a number of different things like muscle development, strength, weight cut, and so on. And if you look at 10 or 20 factors that might influence your muscle mass when you're training, you would have needed to do 30 or 40 experiments to do a good design. That takes quite a long time, and you need a lot of fundings for that. So what we did is, we said there are so many experiments done within sport nutrition. What if we can we do the design, we set up the experiments that needs to be done, and then we look at all the experiments done out there and try to see if we find something by coincident match this point here that we need, this one here, and this one here, and this one here. Then we have a design. It's not really as good as doing your own design, but it's still very good because we have also used these other multi-rate methods to map all other confounding factors to eliminate them, or at least not be aware of them. And almost to our surprise, we were able to find very, very many matches. In one study that we will show you here, results from, we found 28 of 32 needed experiments. But you can still do good statistics even if you miss a few. That's no problem. It's just you need to be aware of where you have your weaknesses in the model and where it's strong. So in this study, we looked at what influence the strength in bench press, depending on how you train. And we have done this for other parts of the body as well, and for muscle mass and fat loss and so on. And here in this particular one, we looked at, for instance, creatine, carbohydrates, protein, timing, number of sets, repetitions, frequency, how many times a week you train. Initial strength, very important. Are you beginner or advanced? That makes a lot of difference, how you should train and what results to expect. If you use progression and a few other things. Here you can see the original variables. The red one is just protein, it just happens to be red here, but it doesn't mean anything. The height of these bars means how influence, if they're big, small or large influence. So it's small, something small has small influence, something large has a large influence. And up means positive and down means negative influence. And you can see here that the interaction terms, which are these on this side here, they are larger, very large. Many of them, and they're large compared to the original settings. So interactions plays a crucial place here. They are very crucial to understand. And that explains why a lot of researchers make different conclusions, because none of them have looked at the interactions, because they haven't done the experiments in a way so they can estimate them. But we have done that. I will go through this in each of the chapters. Then I will just mention a few things. You might be skeptical to all this, and which you should be. You are trained as a scientist to be critical to everything. And then, of course, we want to test that this is a correct model, that it works. So you see some measurement here. This is how well it fits. If it's 1, it fits to 100%. But this data is quite easy to get high, even though your model is poor. You can fit it to crap and you will get a good fit, but it is to crap, so it predicts like crap. This blue one here is actually showing how well your model predicts new data that aren't in the model. Like if someone would call me and say, we're going to do this study, they will get this type of nutrition, they will train like this. What do you think about that? I can answer what the outcome will be by putting it into this model and calculate the result. And 75%, you see here it's about 75 or 0.75. 75% of the variation in the studies we've looked at can be predicted by the model. 25% obviously then cannot. They're part of the noise. What does that mean? Well, for instance, genetic variation, individual variations. We have not looked at that because we don't have the genetic makeup of each individual in these studies. If you would have that, which is an area of research right now, you can within genomics, we probably will be able to get this up to even more than 75%. But this is still very, very good data, very, very good models. I've seen this for over 15 years now, industry data, real data. And sometimes you work with models that have 30% explanation, 70% noise. But at least you know this is what we found, the rest is noise. So you can work with that and make a conclusion and improve things. So here we have very good models. In almost all cases we have more than 50% explanation in our models. So there are individual variation left still, but a large majority of the variation in this study, for instance, where we looked at strength development, can be explained by looking at what the practitioners are eating and how they train. Then you know if they will become stronger or not and how fast it will go. So very, very powerful when used in the right way. And so we'll go through all these things in each chapter, but then we will refer back to the chapter, so we won't talk about the details behind it every time. You have to watch this chapter to get an understanding for what is different, what is unique. So to conclude, when you look at data, do not satisfy yourself with looking just at the original data, also look at the correlation pattern, look at all the factors at the same time. Otherwise you're missing out a lot of information. When you do experiments, do not change one factor at a time, change them all simultaneously according to a design so that you can back calculate the individual response from each of the factors. If you just change them simultaneously as you want, you will have a mess as well. So you need to do it according to one of these structured ways. Okay, so this is really the secret, so to say, the secret behind our studies and why we have been able to come to so many conclusions as we have, and why no one else has been doing it. Well, of course, some of our conclusions are the same as others, but we also have found a lot of new things, a lot of interaction. We have found out how things are put together, how they are connected. That has not been done before, at least not in sports nutrition. But in most medical fields, it has not really been done very, very much of that. Medical fields, it has not really been done very, very much of that. Okay, so now you know more about how to